The Codebreaker, Jennifer Doudna, Gene Editing and the Future of the Human Race by Walter Isaacson. Picture this. It's spring 2014, and a brilliant scientist named Jennifer Doudna wakes up in a cold sweat, her heart pounding against her ribcage. She just had the kind of nightmare that leaves you staring at the ceiling for hours. In her dream, someone had been eager to learn about her groundbreaking discovery, a tool so powerful it was literally rewriting what it means to be alive. But when she entered the room to meet this curious visitor, her blood ran cold. There, sitting with a notepad like he was ready for a casual chat, was Adolf Hitler. His face looked disturbingly pig-like as he glanced up and said, "I want to understand the uses and implications of this amazing technology you've developed." She bolted awake, shaken to her core. Here's the thing about that nightmare. It wasn't just random anxiety. Doudna had helped unlock one of nature's most closely guarded secrets, a tool capable of rewriting the very code of life. On one hand, it promised to cure diseases that had caused untold suffering for generations. On the other hand, well, her nightmare painted a pretty clear picture of what could go wrong. In the wrong hands, this technology could be used to design a future that wasn't just enhanced, but terrifyingly selective. It was basically handing a toolbox to a future Frankenstein. So what was this technology that could inspire such wild hope and such bone-deep fear at the same time? Today we're diving into one of the most important books of our time, The Codebreaker, by Walter Isaacson. And trust me, this isn't your typical dry science textbook. It's a thrilling story about human curiosity, cutthroat competition, and the massive weight of responsibility that comes with discovering how to, well, play God. It's the story of CRISPR, a revolutionary gene editing tool that's handed humanity the remote control to its own evolution. Isaacson, who's previously brought us inside the minds of Steve Jobs and Leonardo da Vinci, now turns his storytelling genius to Jennifer Doudna, the brilliant, driven, and deeply thoughtful scientist at the center of this revolution. We'll follow her journey from a lonely kid in Hawaii, where simple curiosity about the natural world set everything in motion, to the global stage where she found herself wrestling with the moral implications of her own invention. By the time we're done here, you'll understand the fundamental secret that governs all life on Earth. You'll see how a humble defense mechanism in bacteria became the most powerful biological tool ever created. And you'll be ready to tackle one of the biggest questions of the 21st century. Now that we can rewrite the code of life, should we? Let's jump in. Our story doesn't start in some high-tech laboratory with beeping machines and people in white coats. Nope. It begins on the rugged lava rock shores of Hilo, Hawaii, where a young Jennifer Doudna grew up feeling like she didn't quite fit in. Tall, blonde, and lanky, she stuck out like a sore thumb among the other kids, and they made absolutely sure she knew it. They called her haole, a term for non-natives, and the teasing pushed her away from the playground and toward something else entirely: books, and, more importantly, the incredible natural world around her. She found her happy place exploring the lush, wild biology of the island. She became fascinated by eyeless spiders living in lava flow caves and a thorny vine called sleeping grass, whose leaves would curl up the instant you touched them. But here's what set her apart. She wasn't content to just look at these wonders and move on. A deeper question burned inside her. Why? What makes those leaves close? How did that spider evolve? This relentless curiosity, this need to understand the hidden machinery behind life's magic tricks, became the engine that would drive her entire career. The first major lightbulb moment came on a rainy Saturday afternoon when she was in 6th grade. Her father, a literature professor, had casually left a used paperback on her bed. It was James Watson's famous book, The Double Helix. Doudna thought it was a detective story. And honestly, she wasn't entirely wrong. She devoured the tale of ambition, rivalry, and brilliant insight as Watson and Francis Crick raced to crack the code of DNA's structure. But something else in that story hit her even harder. There was a female scientist named Rosalind Franklin whose critical data had been used by Watson and Crick. Watson's portrayal of her was pretty condescending, but young Jennifer saw something completely different. It was like someone had turned on a light. Women could be great scientists. More than that, the book revealed a truth that would shape everything she did. The shape of a molecule determines what it does. Life wasn't some mysterious, untouchable force. It was a physical mechanism. The twists and folds of tiny molecules inside our cells controlled everything. To understand life, you had to understand its architecture. That detective story showed her you could actually hunt down the reasons why nature worked the way it did. This realization put her on a collision course with destiny. When she told her high school guidance counselor she wanted to study chemistry in college, he just shook his head and said flatly, "Girls don't do science." If he'd known Jennifer at all, he would have realized that was exactly the wrong thing to say. It only strengthened her resolve. She would show him she would absolutely do science. And boy did she ever. She went to Pomona College and then to Harvard for her PhD, and it was at Harvard that she made a choice that would change everything. While the rest of the scientific world was obsessed with DNA, the rockstar molecule of life, Doudna chose to study its less glamorous cousin, RNA. Think of DNA as the master blueprint of life, safely locked away in the nucleus of a cell like the original copy of an important document. RNA is the tireless worker who takes those instructions and carries them out into the world. DNA holds the information, but RNA does the actual work. It's a messenger, a builder, and as Doudna would discover, so much more. At the time, most scientists saw RNA as pretty boring, just a delivery service for DNA's orders. But Doudna's mentor, a young genius at Harvard named Jack Szostak, saw things differently. He believed RNA held the key to one of life's biggest mysteries, how life began in the first place. He was captivated by a recent discovery that some RNA molecules, called ribozymes, could actually act as enzymes, those protein catalysts that spark chemical reactions. This was huge. If RNA could both store information like DNA and catalyze reactions like a protein, maybe it was the original molecule of life. This was the RNA world hypothesis, and Szostak invited Doudna to help prove it. Could an RNA molecule actually copy itself? For Doudna, this was exactly the kind of challenge she craved. It was a chance to work on a frontier that was a little neglected but incredibly exciting. It reinforced a lesson she'd learned playing soccer as a kid. Look for the position where you can fill a role that few others can. While everyone else was racing to map the human genome, Doudna dove deep into the mysterious world of RNA. Her doctoral work was what scientists call a technical tour de force. She actually engineered a ribozyme that could stitch together a copy of itself. She was becoming a rising star in a small but profound field. To truly understand how these ribozymes worked their magic, she knew she had to actually see them. She needed to map their three-dimensional structure, atom by atom, just like Franklin, Watson, and Crick had done for DNA. This quest led her to Thomas Keek's lab in Colorado. Keek was a Nobel laureate who had co-discovered ribozymes. There, In what many colleagues thought was a completely crazy venture, she set out to crystallize a large RNA molecule and map its structure, something that had stumped scientists for 20 years. The work was brutal, filled with years of frustrating dead ends and setbacks. But in the fall of 1995, as she was flying back and forth to Hawaii to be with her dying father, the breakthrough finally came. An electron density map arrived from her lab that was so clear she could finally see it. The intricate, beautiful, three-dimensional shape of the RNA molecule folded up like a piece of molecular origami. She walked into her father's bedroom and showed him the image. He was weak, but his scientific curiosity was as sharp as ever. "What does it mean?" he asked. She explained how the shape of the molecule, with a core of metal ions at its heart, allowed it to perform its chemical tricks. In these final conversations with the man who had first placed that detective story on her bed, she was able to clarify her own thinking. He had given her the gift of curiosity, and she was now using it to uncover one of life's deepest secrets. The structure she and her team discovered explained how RNA could be an enzyme, able to slice, splice, and copy itself. It was the beginning of her quest to turn basic science about RNA into something that could genuinely change the world. By 2006, Doudna was a star professor at the University of California, Berkeley. Her lab was world-renowned for RNA structure research and a related phenomenon called RNA interference, a natural process where small RNA molecules can silence genes. She was at the absolute top of her field. And then she got a phone call that would change everything. The call came from a colleague at Berkeley, a microbiologist named Gillian Banfield. Banfield studied tiny organisms in extreme environments, like super salty lakes and acidic mine drainage. In the DNA of these microbes, she kept finding this weird, repeating pattern that she couldn't figure out. The scientific community had recently given it a name: CRISPR. Doudna had never heard of it. She actually thought Banfield was saying CRISPR, like the vegetable drawer in your refrigerator. They met for tea at the Free Speech Movement Cafe on campus. Banfield spread out pages of data, practically vibrating with excitement. She explained that CRISPR stood for Clustered Regularly Interspaced Short Palindromic Repeats. It was this bizarre feature in bacterial DNA. Identical repeating sequences were scattered between unique sequences called spacers. What was it for? Banfield had a theory, one that was just starting to bubble up in the scientific community, thanks to a quiet, obsessive Spanish scientist named Francisco Mojica. Mojica had first noticed these repeats way back in the 1990s while studying salt-loving microbes. For over a decade, he painstakingly tried to crack the code of their purpose. One day, while on a vacation he didn't even want to take, he was in his lab running these spacer sequences through a database. And he found a stunning match. The spacer DNA in the bacteria was identical to the DNA of viruses known to attack those bacteria. It was an earth-shattering realization. He actually exclaimed, "Oh my goodness!" The CRISPR system was an immune system for bacteria. It was a genetic scrapbook of past infections. When a virus attacked, the bacteria that survived would literally grab a snippet of the invader's DNA and weave it into their own genome as a new spacer. It was like taking a mugshot of the criminal. This mugshot would then be used to recognize and destroy the virus if it ever dared to attack again. It was an adaptive immune system that could learn and evolve. The beauty and elegance of this natural defense system actually brought Mojica to tears. Here was the answer to a battle that had been raging for billions of years: the endless war between bacteria and the viruses that hunt them. This was the story Banfield shared with Doudna over tea. The time was right, she argued, for a biochemist and structural biologist to jump in. The microbiologists had figured out the theory, but nobody had actually isolated the molecular components of the CRISPR system to understand how they actually worked. Doudna was hooked. Here was a mysterious system that probably involved RNA, her specialty. It was a brand new frontier, a risky project, and that was exactly the kind of challenge that got her blood pumping. She decided to dive in. And so the stage was set for one of the great scientific partnerships of our time. In the spring of 2011, at a conference in Puerto Rico, Doudna was introduced to Emmanuelle Charpentier. Charpentier was a French microbiologist, a brilliant and somewhat nomadic scientist, who had worked in labs all over the world. She was intense, stylish, and had a sly sense of humor. Doudna liked her immediately. Charpentier, it turned out, was also studying the CRISPR system. She'd been focused on a particular type found in the bacteria that causes strep throat. This system involved a powerful DNA-cutting enzyme called Cas9. But she'd also discovered a mysterious new molecule involved in the process. Another piece of RNA, she called TRACR RNA, short for transactivating CRISPR RNA. She'd figured out that this TRACR RNA was essential for preparing the mugshot RNA that guided the Cas9 enzyme. But she had a hunch it did even more. To figure out the full mechanism, she needed a structural biologist, someone who could help her understand the molecular machinery at work. She needed Jennifer Doudna. As they strolled along the cobblestone streets of Old San Juan, they hatched their plan. They would combine their expertise. Doudna's lab in Berkeley would team up with Charpentier's lab, then in Sweden, to figure out exactly how the CRISPR-Cas9 system worked. The collaboration was a whirlwind. It became a 24-hour operation spanning continents, conducted over Skype and Dropbox. The key experiments fell to two brilliant postdocs, Martin Ginnick in Doudna's lab and Krzysztof Czerlinski in Charpentier's. Their goal was deceptively simple, but incredibly difficult: recreate the bacterial immune system in a test tube. For months, they hit wall after wall. They'd put the Cas9 enzyme in a test tube with a target piece of DNA and the guide RNA. But absolutely nothing happened. The Cas9 enzyme just sat there. Then they tried adding Charpentier's mystery molecule, the TRACR RNA. And boom, it worked. It was the missing puzzle piece. The TRACR RNA, they discovered, wasn't just involved in preparing the guide RNA. It was essential for the final attack. It acted like a molecular scaffold, locking the guide RNA onto the Cas9 enzyme and holding the whole complex steady as it latched onto the target DNA. With the TRACR RNA present, the Cas9 enzyme became a heat-seeking missile, slicing the viral DNA with incredible precision. One evening, soon after this breakthrough, Doudna was at home making spaghetti. Watching the pasta strands swirl in the boiling water, her mind drifted to their discovery. She explained it to her 9-year-old son, Andy. Over billions of years, she told him, bacteria had evolved this totally weird and amazing way to fight off viruses. And it was adaptable. Every time a new virus showed up, the bacteria learned how to recognize it and fight back. It was a moment of double joy for her. The thrill of a fundamental discovery, and the simple pleasure of sharing that wonder with her kid. But the biggest breakthrough was still ahead. As Ginnick and Doudna stood at a whiteboard in her office one day, sketching out the structure of the two RNA molecules, the guide RNA and the trach RNA, they had a light bulb moment. What if they could simplify the whole system? They noticed that the two separate RNA molecules were intertwined. Could they fuse them together into a single, programmable piece of RNA? It was a bold idea. They weren't just discovering something in nature anymore. They were inventing something brand new. They engineered a single guide RNA that combined the essential parts of both molecules. It contained the address sequence that would guide the enzyme to a specific spot on a strand of DNA, and the handle structure that would grab onto the Cas9 protein. When they tested it in the test tube, it worked like a charm. This was the real "oh my God" moment. They paused and just looked at each other. They hadn't just figured out a bacterial immune system. They'd created a programmable gene editing tool. By simply changing the address in the guide RNA, you could send the Cas9 enzyme to cut any strand of DNA from any organism at any location you wanted. It was like a biological version of find and replace for the code of life. It was precise, it was cheap, and it was ridiculously easy to use. They realized they'd developed a way to rewrite the code of life itself. In June 2012, Doudna and Charpentier published their landmark paper in the journal Science. They laid out the mechanism of CRISPR-Cas9 and, crucially, described their invention of the single guide RNA. They knew it would usher in a new era in biology. What they didn't fully realize was that their discovery would ignite one of the fiercest scientific races of modern times. The massive question hanging in the air was this: their system worked beautifully in a test tube, but would it work in actual human cells? Answering that question became an absolute frenzy, a high-stakes competition. While Doudna's lab was packed with brilliant biochemists, they had almost no experience working with human cells. They were entering a race where other labs had a serious head start. Two main rivals emerged, both based in the biotech hotbed of Cambridge, Massachusetts. The first was George Church, a brilliant, eccentric, wild-man professor at Harvard with a flowing beard and a visionary's gleam in his eye. He looked like a cross between Charles Darwin and a woolly mammoth, an animal he was actually trying to bring back to life using gene editing. He was a maverick who immediately saw CRISPR's power and, in his typically open and collaborative style, emailed Doudna to tell her he was going to try getting it to work in human cells. The second rival was far more formidable. His name was Feng Zhang, a quiet, charming, and fiercely ambitious young prodigy at the Broad Institute of MIT and Harvard. Zhang had been hunting for a better gene editing tool for years. He'd read the early papers on CRISPR back in 2011 and had been secretly working ever since to harness it for human cells. When Doudna and Charpentier's paper dropped, it handed him the final, crucial pieces of the puzzle, including the full role of the TRACR RNA and the elegant idea of a single guide RNA. The race was officially on. For 6 frenzied months, these 3 labs worked in parallel, each trying to be first to publish. It was a photo finish. In January 2013, in the exact same issue of Science, both Feng Zhang's and George Church's labs published papers showing they'd successfully used CRISPR-Cas9 to edit genes inside human cells. Doudna's lab, which had to climb a steep learning curve, published its own successful results just 3 weeks later. The line had been crossed. The tool was no longer just a curiosity in a test tube. It was a technology that could alter our own species. And with that power came incredible promise, but also Jennifer Doudna's nightmare, made real. The world was about to learn just how quickly a powerful tool could be used not just for good, but for glory and recklessness. In November 2018, the scientific community was absolutely rocked by a bombshell announcement. A young, ambitious, and media-savvy Chinese scientist named He Jiankui declared that he'd done what was once unthinkable. He'd created the world's first gene-edited babies. The twin girls, given the pseudonyms Lulu and Nana, had been born a few weeks earlier. Using CRISPR, Jiankui had edited their embryos to disable a gene called CCR5. His stated goal was to make them immune to HIV, the virus their father carried. But in his rush to claim the first trophy, he'd broken every ethical rule in the book. The procedure wasn't even medically necessary. There were far simpler, safer ways to prevent HIV transmission. The science itself was sloppy, with evidence of unintended off-target edits scattered through the genome, and the consent he got from the parents was deeply questionable. The news broke just days before the Second International Summit on Human Genome Editing in Hong Kong, where Doudna was one of the organizers. The summit was thrown into absolute chaos. Jiankui, who'd apparently planned to be celebrated as a hero, suddenly found himself the villain of global science. Doudna, David Baltimore, and the other leaders scrambled to manage the crisis. In a dramatic, packed auditorium, Jiankui presented his work. He was defiant, yet seemed almost naively unaware of the firestorm he'd created. He genuinely believed that once he'd succeeded, everyone would recognize his work as a great achievement. Instead, the backlash was swift and universal. Scientists around the world, including more than 100 in China, condemned his work as wildly irresponsible and a gross violation of medical ethics. He'd crossed a line that many had hoped would remain untouched for decades. He'd performed germline editing, making changes to human embryos that would be passed down to all future generations, forever altering the human gene pool. After his presentation, He Jiankui vanished from public view. He was later sentenced by a Chinese court to 3 years in prison for illegal medical practice. The birth of the CRISPR babies was a massive wake-up call. This wasn't a theoretical debate anymore. The power to engineer the human race was here, right now. Doudna found herself at the absolute center of this moral storm. The very tool she'd helped create had been used in a way that genuinely horrified her. For years, she and others had called for a "prudent path forward," urging caution and public debate. But Jiankui had shown that a single ambitious researcher could leapfrog all the guidelines and just do it. The incident forced a global reckoning with some of the most profound questions humanity has ever faced. Where do we draw the line between treating disease and enhancing human traits? If we can eliminate Huntington's disease or sickle cell anemia, shouldn't we? But what about making our children taller, stronger, or smarter? If these tools are only available to the wealthy, do we risk creating a genetic divide? A new class system literally encoded in our DNA? Doudna's own ethical stance evolved. Initially, she was instinctively against any form of germline editing. But, as she heard heartbreaking stories from families devastated by genetic diseases, her position softened. She came to believe that it would actually be immoral not to pursue gene editing to relieve human suffering, as long as it was medically necessary and there were no good alternatives. But she remained firmly against using it for enhancements. No designer babies picking eye color and IQ scores. While these heavy ethical debates raged on, the therapeutic promise of CRISPR started becoming reality. In 2019, a Mississippi woman named Victoria Gray, who'd suffered from debilitating sickle cell disease her entire life, became the first person in the U.S. to be treated with a CRISPR-based therapy. Doctors took her blood stem cells, used CRISPR to edit the faulty gene, and infused the corrected cells back into her body. A year later, she was thriving, completely free of the pain that had plagued her for decades. It was a stunning victory for science and for humanity. Similar trials showed real promise in treating a form of genetic blindness and in fighting cancer. And then, in early 2020, the entire world changed. A new, deadly coronavirus, SARS-CoV-2, swept across the globe, bringing normal life to a grinding halt. The pandemic was a terrifying reminder of just how vulnerable we still are to viruses. But it also became a showcase for the incredible speed and power of the new life science revolution. Jennifer Doudna felt a call to action. As her university at Berkeley shut down, she rallied her colleagues. The government's response was fumbling. It was time for scientists to step up. She converted her research institute into a pop-up COVID testing lab, a feat that would normally take months, but was pulled off in a matter of days. But more profoundly, the pandemic brought the power of CRISPR full circle. The very system that bacteria use to detect and destroy viruses was now being repurposed by humans to do exactly the same thing. Doudna's startup company, Mammoth Biosciences, and Feng Jiang's company, Sherlock Biosciences, found themselves in another race, but this time not for patents or prestige, but to create a rapid, cheap, CRISPR-based diagnostic test for the coronavirus. By programming a guide RNA to find the genetic signature of SARS-CoV-2, they created tests that could deliver results in under an hour, potentially on a simple paper strip you could use at home. And the story of RNA, that humble molecule Doudna had dedicated her career to understanding, took center stage once again. The fastest and most effective vaccines developed to fight COVID were a revolutionary new type, mRNA vaccines. Instead of injecting a weakened virus, these vaccines delivered a snippet of messenger RNA that instructed our own cells to produce a piece of the virus's spike protein. This trained our immune system to recognize and fight off the real virus when it showed up. It was a lightning-fast triumph, built on decades of basic, curiosity-driven research into how RNA actually works. The invention of easily reprogrammable vaccines and CRISPR-based cures meant that the evolutionary balance between humans and viruses had suddenly, dramatically shifted. We may never have to face a pandemic completely defenseless again. In October 2020, as the world was still reeling from the pandemic, Jennifer Doudna was jolted awake by a 3 a.m. phone call. It was a reporter. "I wanted your comment on the Nobel," she said. "Who won?" Doudna asked, still groggy and confused. The reporter was stunned. "You mean you haven't heard?" "You and Emmanuelle Charpentier. The Nobel Prize in Chemistry was awarded to them for their development of a method for genome editing. It was a historic moment. The first time a science Nobel had been won by two women together. It was recognition not just of their brilliant discovery, but of the dawning of a new age, the age of humanity's ability to rewrite the code of life itself. Reading the codebreaker feels like watching the birth of a new world in real time. Walter Isaacson doesn't just hand us a biography of a scientist. He gives us front-row seats to a revolution that's going to shape the entire course of human history. There are three big lessons I think this story holds for all of us. First, it's a powerful reminder of why pure, curiosity-driven research matters so much. Jennifer Doudna didn't wake up one day thinking, "I'm going to invent a gene editing tool," or "I'm going to help create a COVID test." She was simply a girl from Hawaii who was intensely curious about why a leaf curls when you touch it. Her entire career was powered by a desire to understand how nature works at its most fundamental level. She studied RNA because she found it beautiful and mysterious. That basic, unguided curiosity is what led her to CRISPR. The whole story, from yogurt makers to Francisco Mojica to all the scientists who pieced this puzzle together, reminds us that the greatest innovations often spring not from chasing a specific application, but from the simple, joyful pursuit of knowledge for its own sake. Basic science is the seed that grows into technological breakthroughs. Second, this book is a masterclass in the power of collaboration and the productive tension of competition. The discovery of CRISPR was absolutely a global team sport. It involved a Spanish microbiologist, French and American food scientists, a Parisian wanderer working in Sweden, a Czech researcher in California, and dozens of others. The breakthrough moment happened because Doudna and Charpentier chose to combine their strengths instead of working alone. Yet the story is also packed with intense rivalries, brutal patent battles, and races to publish that were fueled by ego and ambition. This isn't a contradiction. It's just how science actually moves forward. Competition accelerates the pace of discovery, but collaboration is what makes the biggest leaps possible. The creative tension between the two is what drives progress. Finally, and most importantly, The Codebreaker is a deep meditation on responsibility. With the power to rewrite our own genetic code, we've become the authors of our own evolutionary story. Doudna's Hitler nightmare wasn't just a random dream. It was a warning about the heavy burden that now rests on all our shoulders. The story of He Jiankui shows us this power is already being used, and we absolutely cannot put the genie back in the bottle. We're all stakeholders in this conversation now. It's not a discussion that can be left to scientists in their labs or ethicists in their offices. It belongs to all of us. What kind of future do we want to build? What does it mean to be human in an age where our very biology is becoming a matter of choice? These aren't questions for science fiction anymore. They're the defining questions of our time. So here's a simple thing you can take from this. The next time you see something in the natural world that sparks your curiosity, a lizard changing color, a sunset painting the sky, water beating on a leaf, don't just glance and move on. Pause and ask why. Ask how it works. That simple act of engaging your curiosity is the first step on the path Jennifer Doudna took. It's the engine that drives all discovery, and it's the starting point for understanding our place in the magnificent, intricate code of life. That was a summary of The Codebreaker by Walter Isaacson. We've explored the main currents of this incredible story today, but I'll be honest with you, a summary can only ever be a map. The real treasure is found when you make the journey yourself. The true magic of this book is in the details, the human stories, the scientific rivalries, the personal struggles, and the quiet moments of awe that Isaacson captures so brilliantly. This summary is designed to help you decide if this journey is for you. If anything we've discussed today has sparked your own curiosity, I really encourage you to grab a copy. Reading it is more than just learning. It's an experience that will fundamentally change how you view life, science, and the future of our species. The link to the book is in the description. Also, if you found this journey valuable, please take a moment to like this video and subscribe to the Billionaire's Bookshelf. It helps us reach more curious minds like yours, and make sure you won't miss our next exploration into another book that can change your life. Let's keep learning and growing together.